The professor became Professor of Haematology at the Medical University in Nantes? Nantes.
Great.
Sorry, my French is really bad. Back in 2010. He's also in charge of the lymphoma research program and early phase clinical investigation in the haematology unit. He gained his PhD in the specialisation in blood cell therapies, has a very special interest in mantle cell lymphoma, but obviously does cover all types of lymphomas, and is a member of the Lymphoma Study Association, where he's co-chair at the Mantle Cell Lymphoma Committee, and an expert in the treatment of mantle cell lymphoma. And he's participated in the European Society for Medical Oncology consensus and the guidelines of treatment for mantle cell lymphoma, and obviously conducting several clinical trials. So welcome, Stephen, and I'll just quickly give an update on Michael as well. They'll both be working quite interactively, as I said before. So Michael is the lead of aggressive lymphoma disease group within the clinical haematology unit here at Peter Mac and Royal Melbourne Hospital. He specialises as the principal investigator in phase 1-2 clinical trials of clinical anti-cancer drugs, with a current focus on aggressive lymphomas and novel immunotherapies. However, his experiences across all types of haematological malignancies as well. And he's recently succeeded in developing and commencing CAR T-cell program here at Peter Mac in various trials. And Michael's also an active member of our Lymphoma Medical Advisory Committee too, so I'm lucky to be working with him quite a bit. So please join me in welcoming both Michael and Stephen.
Thanks, Donna. Just bear with me while we just connect up the technology. So I'm delighted to have Stephen here because some of the work that he's done in France has changed the standard of care.
Take me off.
Excuse me. For mantle cell lymphoma in particular, but Stephen's also been involved in developing novel therapies in lymphoma, and his work really has influenced how we treat lymphoma, both in the frontline setting and in the relapsed refractory setting. Excuse me, I've got a 1.5-year-old and she's taking her toll. And I just woke up this morning and realised that my voice is going. Excuse me, so excuse that. So keep your distance from me this morning. I'll keep a good radius from all of you. We're going to talk a little bit about the standard classification of lymphoma, basically talk a little bit about frontline treatment, but then really want to focus on emerging treatment. Donna's got a few questions she wants to ask, but in addition, we'd be really keen in hearing questions from you about specific things. We're just going to provide a bit of a framework initially, and then I'll talk specifically about CAR T because I was asked to speak about that. But we'll talk initially about the framework, novel therapies. We'll have a bit of a chat, and then I'll come back and talk a little bit about CAR Ts, what the current state of play is in the Australian context. Stephen works in France, of course, and so is able to tell us special things about the European experience. There are just more people in Europe, which means that you can do bigger trials and answer bigger questions, which is really part of the expertise that he brings. And so he'll obviously not be aware of the Australian landscape. And so anything he says about current treatment of lymphoma and emerging therapies, I can try to give you an Australian context. So there'll be a bit of to and fro, and so feel free to interrupt and ask questions as you see fit. Thanks very much, Stephen.
Thanks, Mike. Good morning to all of you. It's a real pleasure to be here in the Peter Mac, in this wonderful building that is really devoted to research in cancer and lymphoma in particular. So as has been mentioned previously, I come from France. I work in a city called Nantes. It's on the west coast, on the Atlantic Ocean. Just you will, for those who knows, or maybe when you get back home, you will look on the map where it's located. It's a 500,000 inhabitants cities, and I work in an area of 1.1 million inhabitants. So it's a university hospital, meaning that we treat patients, we do research, and we teach. These are the three reasons why there are hospital universities. And we are working in France in a network. You mentioned the name of our cooperative group. It's called LISA, Lymphoma Study Association, in English. It's all the big French centers are working together in this cooperative group. It's an academic group. It's devoted only to lymphoma, and we have connection and with colleagues in Belgium, for example, who are also members of the LISA group. But one important thing to mention is that is not treating lymphoma is not something you do on your own, in your own institution and in your office. It's important to have collaborations with colleagues all over the world. So there are European collaborations, and we have collaborations with colleagues in Australia. It's not my first time in Australia, and we see each other frequently, so it's a good opportunity for us to discuss about the new treatment, about our new ideas, and sometimes we build together clinical trials at the international and the world level, and I think it's an important progress that had been made these last years is to be able to collaborate at the large level, because if lymphoma is not a rare entity, there are many lymphoma entities, and it's sometimes very difficult to build large clinical trials. If you do it on your own, you need colleagues all around the world to participate and to enroll patients into clinical trial, which is a major challenge today, is to run these clinical trials. Maybe we could have a few words at the end of the talk about the major issue for the clinicians to build these clinical trials. And I think today it's really something we have to work with patients association is how to facilitate the way we can build trials to move forward safely and to try to do it for a reasonable price. It's a major challenge, and I will be happy to talk to you. One of the problem in lymphoma is that there is not one lymphoma, as you know. There are a lot of lymphoma entities, and it's difficult to explain to the patient that your case is completely different from another one's. It's a lymphoma, but your lymphoma entity is not the same than the other lymphoma entity. So there are wide range of treatment from wait and watch to autologous stem cell transplant frontline. And I guess it's the same in Australia. Sometimes when you come to see your doctors, you have to wait a little bit before your consultancy, so patients discuss together and they don't understand why their lymphoma is treating this way, while the other lymphoma is not treating the same way. And what it's important to keep in mind is that this definition of each lymphoma, it's a huge work. It's not something that is defined locally. This book is a very important book. This is the first page of the book. It's called the WHO classification. This is a big work that classifies and defines each hematological malignancy, not only lymphoma, but also leukemia, chronic myeloid leukemia. All hematological diseases are in this book. It's a 436-page book, so it's something hard to read. It's not as entertaining as Game of Thrones, believe me. It's very, very long to read, and it's purely definitions. How it looks like, what the phenotype, are the genetic abnormalities, and why you can call it follicular lymphoma, mantle cell lymphoma, or DLBCL. It's a work that is repeatedly updated. It's not the first version, this is the last version, as there have been several versions before, not because we are discovering new disease, but because we requalified the disease, because we get more and more advanced new technologies, and these new technologies change the way we see the disease. So we are not discovering a new disease, we are better describing a new disease. It's like when you look at the skies, if you are in the Roman Empire, you say this is a god or a star, etc. But once after times, you get better technology, and then you see a star, and this is the constellations, and this is different things, and everything changed with the advance of the technology. And this is why these books need to be updated. The recent version introduced a new, very important concept, which is the molecular background of the tumor. It's not only description of the cell, description of the tissue, description of protein on the surface of the tumor cells. It's also what's happened at the DNA level, what are the abnormality. So this is getting more and more precise, divided, and one key example is DLBCL, diffuse large B-cell lymphoma, which is a group of disease that is very heterogeneous. And now that we get new technology, we better understand why there are differences between this DLBCL and this one. It's because we find new characteristics in a subgroup of DLBCL. It's important to keep these things in mind because it helps us to better understand the biology of the disease. So it means we have a better view to the future treatment of this disease because we better understand why these cells behave wrong way. The other advantage of this kind of description is it's like a language. This is the only way for us, Mike and I, to speak about the same disease, and we understand and we clearly talk what is a follicular lymphoma is my mind, is what is a lymphoma is the same in his mind. And when you live on the other side of the world, it's not easy to agree about the same definition. And thanks to this book, we all around the world use the same terms to define the same disease. So if we build clinical trials together, we will define the disease the same way, because or thanks to this book, right? And in this book, you will find that there are not one, but many tumor hematological malignancies and many lymphomas. I didn't count, but it must be more than 80 different lymphoma entities. So it's big numbers of disease. Some are really very rare. You may see some like 5 or 10 patients in your own career, and others are more frequent. So this is a big advantage because we better understand the disease, we better understand its biology, but it has also some disadvantage. It's difficult to build clinical trials with small disease. Phase 3 is almost impossible to build if you only see 1 or 2 patients per year or 1 or 2 patients every 10 years. So the more we get, the more we characterize the disease. On the other hand, we pay the price of the difficulty to build new clinical trials. So it's also a challenge for us clinicians who do clinical research to also change the way we are doing clinical research. The big standard is called phase 3. It's treatment A versus treatment B, and you see which one is the best. But then you need a lot of patients. When I mean a lot, it's hundreds of patients. For some disease, it's almost impossible to do it this way. And if you divide the disease into small entities, it's getting more and more difficult to do these phase 3 trials. So we are more precise, we have targeted therapy, but it's also a challenge for us to build clinical trials that will demonstrate it or not the interest of one drug in this disease. And it's also a challenge for the authority who will at the end pay the drug or to admit that its drug is active in this disease. So it's a huge challenge, and I think nowadays it's one of the most important things, a problem that we are facing is a lot of new drugs, but a lot of new entities. Drugs are getting more and more, the price is rising up like crazy with new molecules. How can we do to demonstrate that one molecule is active or not in a disease? And today, I think no one has a clear answer about these questions. And this is something we will have to think about. So a lot of disease, well-defined disease, a lot of new drugs, that means a lot of questions in the future. However, when we discuss together, there are big blockbusters in lymphoma. This is the way it can be divided in 3 pieces. And it's the way we divided lymphoma in the 90s. There were indolent lymphoma, aggressive lymphoma, and let's say very aggressive lymphoma. This is the way lymphoma was defined in the 90s, and it's based not on molecular definition, even not cytologic definition, it's based on clinical definition. An indolent lymphoma is a lymphoma that takes a lot of time to grow up, 10 years, 20 years, and sometimes you don't need any treatment. But the term of indolent, I mean, when you, the patients, you don't think that your disease is somehow indolent. It has nothing to do with, you prefer not to have any disease, and our cancer can be indolent. So this is not a perfect way to describe, but to divide the disease into different behavior. So you have the indolent lymphoma, like follicular lymphoma. It's a typical indolent lymphoma. Then you have the high grade or aggressive. The typical, the classic entities is the diffuse large B-cell lymphoma. It's an aggressive because if you don't treat the patients, the tumor grows very fast, and then you see the clinical impact of the tumor on the quality of life of the patients, and it grew up in a very, very short period of time because the cells are cycling a lot and they do proliferate. And you have some kind of super aggressive disease, and this entity is something that has been better understand these last 10 years. These are diffuse large B-cell lymphoma that are very aggressive because they carry cytogenetic abnormalities that completely change the behavior of these tumor cells from aggressive to very aggressive. It's like in a computer, you get your programs. In the tumor cells, there is a big problem in the program, and aggressive has a very big problem in the program. The cells proliferate like crazy, and these cells have major abnormality targeting key genes like MYC genes, for example, and these cells behave are cycling a lot, and of course, this has major impact on the quality of life. So this classification is a very clinical classification. It is not used in clinical trials, but it's still something that we use. We still use these terms in our daily practice because it makes sense from a clinical point of view. An indolent lymphoma. If you look at this book, you will not see any definition of indolent, because it's not the purpose of this book to describe the clinical behavior in the tumor. This book does not describe the tumor based on the clinical point of view, but from a biological point of view, right? But we use both terms in daily practice. It's a mix of two different approaches. So what is our challenge today? Our challenge in the coming years, is to go one step forward and to better define the entities. Not only because we want to describe every little thing, it's when you describe something precisely, you're able to treat with more accurate drug this tumor. And this is today what we're trying to do in the lab and in clinics. It's to have a perfect picture of the tumor and to be able to select the right drug for this tumor, and most importantly, for these patients. Because in the same tumor, they are heterogeneous. The patients are heterogeneous. The patients are not the same because they got the same disease. They are also different from one patient to another with the same disease. And this makes the world of lymphoma even more complex. Today, we're talking a lot about personalized medicine, about targeted therapies, etc. In clinical practice, it's quite frustrating because sometimes we are not doing targeted therapy. R-CHOP, Hyper-CVAD, or DAIPOC is absolutely not personalized approach. However, it works. That's which is the most important things. So for people like the two of us and all our colleagues in the world today, we have more drugs, but we have more disease. And one of our major challenges is to find a new way to treat patients based on the on our biology knowledge that is improving very fast. So it's fascinating, and sometimes it's also very frustrating to get so many drugs and so many diseases. So in next talk, Mike will talk about one of the most important advances, which is a CAR T cell. And this is another way to treat the patient that it's called immunotherapy. Thank you.
Thanks very much. So we'll go to questions now on what Stephen's just said. He's just provided a very nice overview, and I've got a few questions as well. So does anyone have any questions?
Is there any questions so far?
Sorry.
We're webcasting this, which is why we'd like you to use the microphone so that the people on the
We have a very unique problem in Australia where a lot of people live in remote areas, probably a third of Australia, and many of them to see a haematologist or an oncologist is a day's airline trip away. How do we address this? Most of us, I presume, here come from Melbourne. Melbourne I regard as one of the best places in the world to be treated, but probably a third of Australians are completely remote from diagnosis and effective treatment. And the economic base for that, for them, is poverty-stricken.
What I can tell you is that it's not only an Australian problem. You have a very, very big country, so you may be far away from a big city like Melbourne. But for a French, when you're living 50 kilometers away for a university, you're already far away. So it's very relative, you know. And we have the same issue in France. There are few hematologists, in fact. It's not like cardiologists or like dermatologists. There are few hematologists. So an important point is to create networks between hematologists, and first at time of diagnosis. In France, it's national recommendations once you have a biopsy and that for tumor that may be a lymphoma this biopsy has to be double checked by so-called lymphoma experts at the national level. So we have local pathologic reference that receive the tumor and they confirm or not the diagnosis. This is the first step to get access to the diagnosis. Once you get the diagnosis, I think this is the role of the doctors to refer to a specialist, whatever is the distance. I think there are no perfect way to organize these kind of networks, but we have new technology nowadays that even if you live far away, you're not so isolated. You can have contact with someone from a reference center like the Peter Mac and start to talk about what will be the best treatment for this particular case. But it's a huge challenge for all of us to provide the better treatment and access to even to the diagnosis to all patients.
Yeah, so certainly I agree completely, and I think Stephen really hit on a key point, which is that for every new cancer diagnosis, but particularly in lymphoma, getting a second opinion on the pathology is an important early step, because so much of your success in having your lymphoma treated is the first treatment. Careful selection of the first treatment to avoid relapse as best as you can. And then after that, of course, if a patient has a relapse, you still have the same challenge. And so getting a lymphoma expert in the lab to confirm the diagnosis is a really important equity issue. So if you're from the country, your pathology should still be looked at by someone who works in a big lymphoma centre under the microscope. The other issue is clinical service provision. So it is extremely expensive to provide certain specialist cancer services, and it's also not always a good model to have hundreds of hospitals using some of the most unsafe treatments, because what you'll tend to find is that you get people who do certain treatments very infrequently and are then less able to cope with toxicity if it arises, and that could be an issue for CAR-T, but allogeneic transplantation, autologous stem cell transplantation, even just intensive inpatient chemotherapy are examples where you need to do enough of it to be a good doctor and to be an expert in it. And so there are things like that people just need to travel to have. And if we break it down into too many centres, you're not going to have enough lived expertise. Our approach to the other question around appointments and reviews is telehealth. So we now have a very good web-based video consultation system at Peter Mac, which is really very robust. And I see second opinions, particularly interstate patients now. I would say I see 3 or 4 patients a week that video to me. And we have a video consultation first, where we can talk about whether or not coming to Victoria is a good way to spend time. We have a video consultation about where a second opinion might take someone before we do the very resource-intensive thing of asking someone to travel or sending their tissue down to us for a second opinion or to perform a high-tech genomics test. So I think the Centres of Excellence type model is still a very good one for highly specialised services, and technology will help get us around the tyranny of distance.
Can you also comment a little bit on the multidisciplinary meetings that you probably both have? Do you have a lot of clinicians who are calling in from around the country as well to be able to have expert advice rather than doing a second opinion too?
Yeah, so we use it. Our multidisciplinary team meetings are held in a room like this, small ones, and we have video conferencing facilities. We routinely have people from country Victoria phone in, so we have special hospitals that are connected into us and report all of their patients, and we discuss all of their cases in our group MDT. We have about 45 doctors in the room, and nurses and radiation oncologists, and so a range of specialists as well as the pathologists and imaging doctors. And regional hospitals routinely phone in to us, and some regional hospitals are big enough to run their own. So it just depends really on what the individual needs of a hospital are. I think what you want to know if you're in private medicine is whether the private system is providing that same service, and certainly some of the big private hospitals around the country make a point of doing MDTs, and in my experience they've been very good MDTs as well.
So you know there's a lot of conversation that does go behind the scenes as well. Stephen, do a similar sort of system work in France as well, where and do patients have to travel a long way in France?
It's the same. It's the same system. We have weekly meetings with colleagues working in other institutions around Nantes, and it's mandatory by law in France. You cannot start a new treatment if there is not a multidisciplinary meeting that takes the decision. It's not something you can do by your own in your own office. You have to discuss the case with colleagues from different hematologists, pathologists, radiologists, and etc. And we all together we decide for this patient it will be this treatment. It's mandatory by law for the front line, not for relapse, which I think is a shame because the problem occurs mainly at relapse and not front line. So this is exactly the same in France.
I think it's stricter in France. So you have the European directives, and I don't think that we yet have those directives in Australia, those similar sort of directives. So when I was working in the UK a decade ago, these directives just came in, where every single patient, as you say, by law, you literally couldn't treat a patient. In Australia, you can. And one of the tensions is between discussing 100% of garden variety lymphoma X versus spending more time on a complex new diagnosis. I think the health systems are grappling with that tension in Australia, but there's no doubt that in Victoria, the rules around MDTs are being tightened and actively audited as we speak.
Yeah, it's interesting how different states work quite differently in Australia. It probably works the same in France as well. Yes. Actually, sorry, Sonia, would you like a question first?
Is there any benefit of early treatment of indolent follicular lymphoma with MabThera in long-term survival as opposed to watch and wait? And what facts are considered in early treatment of follicular lymphoma?
Well, it's a complex question. Today, wait and watch is standard of care, but there have been few clinical trials comparing wait and watch and rituximab monotherapy, and the major trial didn't show any difference in terms of overall survival. It's remain local practice. In my institution, for example, we treat indolent follicular lymphoma with 4 infusions of Rituxan. I don't claim that it is better to do it rather than wait and watch. There is no clear demonstration of that. Why we're doing it, it's more because we feel more comfortable to give a treatment that is rather safe with low toxicity. And if we get a chance maybe to prevent from progression of the tumor, this is at the moment of the diagnosis. But it's not based on clinical demonstration. And if a colleague say, I wait and watch in our weekly meeting, no one say, no, you have to treat patient with Rituxan. So
generally, you do the 4 weeks of rituximab
yes,
as a matter of course, generally.
Yes, generally. Yeah, but colleagues in other institutions wait and watch for this patient, and there's nothing to do in this. It's more a local decision. There are things that are not clearly demonstrated, and there is still a part for personal interpretation or personal, let's not say feeling, but experience. Because I have patients who have Richter syndromes a few times after diagnosis, and I'm always wondering, maybe I should have done something at the beginning, and etc. But it's more a personal feeling rather than a clinical demonstration.
One more. If it's stored in wax, the lymph node, like 4 or 5 years ago, is it possible to do new pathology? And also, what biological characteristics lead to progression?
Sorry?
The pathology.
Yeah.
If a lymph node is stored in wax, is that any good still to look at it again if a second opinion wasn't sought? To look at it more detailed or not?
I don't think there is. No, I don't think there is a need. If you have a right diagnosis and nothing really changed, I think there is no need to rebiopsy again.
No, from that sample, I mean. Can you look at that, or is it no use because it's been stored in wax?
So the paraffin-embedded samples last for a very long time. Yeah. And one of the issues can be if someone took lots of slices, that there's sometimes not much left. But if there's something left, if there was a big original biopsy, most hospitals in Australia are obliged to keep, in fact, there's Australian regulation about how long you keep that tissue for. That can be reviewed down the track.
It can be looked at again by another team.
It can be. So 5 years is not too long. Yeah. There are some limitations. So, for example, genomics can be done, but the longer you go, the more error you get in the machine that does the genomics. So it can be a lot more noise if someone's tissue is paraffin fixed and embedded, and then the longer it gets, the noisier the data gets. But it is possible to go back and look at those archival samples if they exist.
Oh, they took a few out, but there's only one apparently because I rang up and they have to keep it for 10 years.
That's right.
But someone told me it's no good because it's in paraffin to look at in the lab.
It's usually fine.
So it's fine. And what characteristics in the pathology lead to transformation? Do we know, or is that more a lab question?
Yeah, so it's a tough one. So that's still a matter for research. There isn't a single molecular marker or immunohistochemical marker, and there's a lot of research around the clinical characteristics, the stage, and how advanced a lymphoma is when it presents, about what the transformation risk might be, but we don't have a good crystal ball for that. We really don't. So we can't sit down with an individual patient and say, oh, look, I think you've got one that might transform in the future. There are molecular markers in follicular lymphoma that use complex genomics and that predict overall outcome. They don't always point to transformation specifically in terms of the overall outcome. And they're not routinely being used in clinic. Do you use the mFLIPI?
No. The problem with this is this mFLIPI or all this biomarker is that they have shown their prognostic value in a large scale of patients and not for personalized medical decision. You see, you can say all these if you have this abnormality, there is 60% risk of Richter syndrome in 10 years. When you're a patient, what does it really mean? 60% or 40% in 10 years?
If it's you, it's 100%.
If you're right. If it's yours, you get 100% or 0%. Yeah. But these systems are built for populations of patients and not for personalized approach, which is a problem. But today we don't have the tools to do better than that.
What you were talking about before, the personalized looking at the
So the pathology review is just about what Stephen was talking about looking down the microscope and making sure that of the several subtypes of lymphoma, this is correct, right? But it doesn't allow, beyond getting the diagnosis right, it doesn't help really predict the future, with some exceptions.
Thank you.
Yeah.
Well, when you're first diagnosed after a biopsy, you're given a category, and in my case it was mantle cell lymphoma. And the oncologist then decided what I would probably regard as a general therapy of chemotherapy and immuno. And I'm just wondering whether, in fact, a deeper analysis or diagnosis of the lymph glands would actually result in a more targeted approach to.
Couldn't be asking a.
Pardon?
I couldn't be asking a better expert on this question. This is
that's
Stephen's
right.
thing.
That's my topic of research is mantle cell lymphoma and the targeted therapy. There are a lot of biologic parameters and you can look at in mantle cell lymphoma like in the other lymphoma entity. All these findings translate into a change of your medical decisions. There is a big bridge to cross before to go from general treatment to targeted therapy. This targeted therapy wording is a little bit, the word targeted therapy is a little bit risky somehow. It's a word that we use a lot, but what does it really mean? Targeted therapy means I know what is the drug and I know where it goes and I know what is the effect of these drugs and that this drug is required for these patients and not for the other one who has the same mantle cell lymphoma entity. To be honest, today, we don't know how to do that. We have new drugs like BTK inhibitors, works really well, but 30% of the patients will not respond to BTK inhibitor. Why? We don't clearly know why. We cannot identify these patients at diagnosis or at relapse and predict that they should or should not receive this drug rather than the other one. It's a technology that we don't have in hands today. And that's what I said, it's quite frustrating because we know that one mantle cell lymphoma is not equal to the other one, but we treat all the patients the same way. It's not because we like it, it's just because we don't know how to do better than that. I think in the close future, whatever I mean close, we will change. But for this, we really need this clinical trial. We'll need cohorts of patients with biopsy of not. And every patient should be included in a survey program where we collect all data about his quality of life, response, his tumor. But then you can imagine the price, the cost of such a trial or such a survey, which is in fact completely needed, but it's a huge thing to do. And it's not only a question of money, it's a question of logistics, it's a question of time, of human resources. So today we have targeted therapy. We know the difference between one patient and another patient, but we don't have the technology to really have a personalized approach, which is a shame, but we're getting more close and closer to this, but it will take a little time.
There's a philosophical decision that we need to make as a community, which is to have a loss leader here. We need to take a hit in terms of cost to describe every patient's lymphoma so that we understand the frequency of the micro subtypes of lymphoma that genomics are going to bring out and tell us about, so that we can then plan towards how to develop and do targeted therapies. And one of the problems we have is if genomics is the answer for splitting up these different lymphoma types and allowing us to pick targeted therapies, how do you sell that to government? Because they sit down and go, tell us about the cost effectiveness of doing genomics in a patient with acute leukemia and how will that change how they go? And you say, well, there's been this randomized study for this particular mutation and if we give them this drug, it will save this many lives. And then government says, fine, we'll pay for that test. But in lymphoma, we're actually years behind that. And that's where, as an organisation, we've said, right, we want to describe the lymphomas as best we can, and we don't know what we're going to do with that information, and there's a risk that we're wasting our time, but let's just learn. And to do that, Peter Mac has had to seek significant philanthropic funding, and we've had some, very supportive donors and organisations to do this, and everyone's taking a risk that it's a waste of time, but we're doing it in the hope that by describing everyone's lymphoma over years, we're better able to contribute to that effort to target therapies. And so the Wilson Genomics Centre here at Peter Mac, we're sequencing everyone's lymphoma and we're not giving reports back to patients routinely. We are just soaking it up, putting it into a big compendium so that at the end of the day, when a company comes along and says, I've got a targeted therapy for EZH2 mutation or something, we can say, well, we've seen this many patients in the last 2 years, which means we'll see probably this many patients in the next 2 years, so you should do that trial here. And that's been our strategic approach, but that's going to take years to actually bear fruit.
Yeah.
And it might not turn out to be the right approach, but that's been our approach.
We also have to be honest and not to oversell our knowledge about the tumor. Yeah. There have been some trials based on tumor cell characteristic. For example, in DLBCL, our colleague in UK make big trial in DLBCL. They characterize the tumor cells, and if you have this characteristic, you were treated with a chemotherapy plus this was ibrutinib, a new drug. They didn't see any difference at the end. So they were a good rationale, a good design. Everything was supposed to be a success, and it wasn't. Why? Because we do not understand all tumor cell, and one abnormality in the tumor is maybe not the one who drives the behavior of the tumor cell itself. So all the connection between all the anomalies, it's a complex pictures that we don't fully understand. It's like a puzzle. We understand a little piece of the puzzle, but we don't have the full pictures or what will be the conclusion. So we have to be very modest about what we really know and understand in the disease and not oversell. And this is the same when you open the radio and read the journal, amazing new targeted therapy, and it's a lot of hope. But sometimes it's more commercial than real science demonstration.
And the opportunity cost is huge. You put 1,000 patients into a trial that fails. Maybe you should have done dozens of other trials that were more exploratory, that for an individual patient you might have been more speculative. So this is a real dilemma.
Yeah, it's clinical trials are. The price of clinical trial is rising up like never. It's getting more and more financially complex to rise clinical trial, and not only because of science. That's the worst things. It's also because we have. It's like someone is putting more hurdles on our way where we try to find something new. I wrote something in British Medical Journal, an article, an editorial called "Bureaucracy is Strangling Clinical Research," and it is something that we're facing every day. That it's getting more and more complex to run clinical trials, and not because the price of the drug is higher, not because government asks more things. It's just because they ask us more and more data about safety, about quality of the data, and it costs a lot of money to get these informations. And for academics just like us, we need sometimes to have very easy questions to ask, and we want to do it fast. But if we spend all our time to get funding for some, then it's a waste of time, a waste of money, and the quality of the clinical trial is not improved because it's more expensive. There is no direct link between the cost of the trial and its efficacy. I run a trial in mantle cell lymphoma called the Lima Trial. It's been published 2 years ago in the New England Journal of Medicine. I started this trial in 2007. I published the result in 2017. It takes 10 years from the first patient in, the first time I write to have the publication. 10 years. It could have been 5 years, but we spent 5 years to get the funding, to do the monitoring, etc., etc. So it's a waste of time. It's a big waste of time. And this trial, the cost of this trial was around, let's say, 3-4 million euros, which is nothing. The same trial now will cost at least 20-25 million euro for exactly the same result at the end. No change, it will be exactly the same, but I will have to face more hurdles based on safety data, monitoring, adverse events reports, and all these things. That will be 70% of the cost of the trial, which I think at the end is a shame and waste of time for everyone, and first of all for the patient. Because at the end, we show that there is an overall survival benefits with the new treatment that we propose. Overall survival benefits, not progression-free survival, but overall survival. To do it today, it will be much more complex to do, much more.
Yes, just talking about clinical trials, I'll just note on the fact sheet that I've got here that the drug obinutuzumab is still under investigation. Now,
I'm having that on a monthly basis with bendamustine. What's the current findings on this immunotherapy?
Obinutuzumab is a follow-on drug from rituximab, so it's an anti-CD20 antibody, which is broadly applicable theoretically to any lymphoma that carries CD20. And the obinutuzumab has been tested for particular lymphomas. And if you've got a drug that targets a whole bunch of lymphomas, you kind of have to pick some. And so essentially what the drug companies do is they go for the most common ones, because that secures essentially a business. So they go for the common ones first. And so the ones they went to first is diffuse large B-cell lymphoma, where there was no advantage, and then follicular lymphoma, where there's a significant advantage with respect to risk of relapse, because it substantially reduces the risk of relapse, but probably at the cost of some increased short-term and medium-term infection risk. So that's Gazyva, and it's being explored in other contexts and in combinations with other lymphomas, and those explorations continue.
Thank you.
Eric.
You knew I was going to ask a question, didn't you?
I did.
Just with the trials, I'm trying to understand if a trial is done in France or in America, can another country automatically adopt it? Or does that country, like say Australia, have to go through another trial mechanism before it gets approved?
For Europe, the standard of approval is based on phase 3. So a phase 3 trial is a standard of care versus a new treatment, the challenger, if you want. EMA, the European Medical Agency, requires this kind of designs for a drug to be approved. When it's approved at the EMA level, every country of Europe has to give its or approval or not, and a price to the drug. Australia or Japan or FDA are free to say yes or no to the trial to a drug. The company has to explain why they want to be approved in this indication based on the trials. But every country, every continent, every organization is free to say, I take it as a demonstration, or I want something different. Let's, for example, ibrutinib, Imbruvica, for mantle cell lymphoma, was first approved in the US based on the phase 2 trial. It was only a phase 2, so no comparison. But the FDA admit that this result was quite outstanding, and there was no need to demonstrate the efficacy of this drug versus something else, because there is no something else. So they approve the drug, and then they get a price. In Europe, this was not sufficient. In Europe, the EMA said, we have a drug that is approved, which is temsirolimus. It's approved for relapsed refractory mantle cell lymphoma. You have to prove me that your drug is better than the one we already approved, which was a stupid trial because temsirolimus doesn't really work. Mm-hmm. But EMA said this is a standard of care, and you didn't demonstrate any benefits. They say yes, but we have a phase 2. Yes, but it's a phase 2. There is no comparator. So I will not allow your drug if you don't do this trial. So in Europe, we repeated the ibrutinib trials versus temsirolimus. This trial demonstrated that ibrutinib was better than temsirolimus, and ibrutinib was then approved. There are countries like Japan that repeat every clinical trials because they believe that the genetic background of the Japanese people is too different from the European Caucasian people. So they want to have the same trial, not phase 3, but sometimes phase 2, just to confirm that what is true in the French is also true in the Japanese. But that's national policy. There is no international organization that can approve a drug worldwide based on one clinical trials.
And approval is not just about efficacy or toxicity, it's also about cost effectiveness.
Yeah.
And so the threshold for cost effectiveness is obviously going to be regional in terms of what that threshold is, and it's going to depend upon the insurance framework, basically.
And in France, but I don't know if it's the same in Australia, you can get approved but have no price.
Exactly.
So
we
if you
have
approve with no price, no prescription.
that. Yeah.
Because the French government have to find an agreement with the pharma company about the price. And if they don't agree, there's no price, means no prescription.
In Australia, it's possible for a drug to be approved but not reimbursed.
That is,
And in that context, there can be a price, but it's down to the patient to pay because the government has not yet reached an agreement that the price is something Medicare will pay for or the Pharmaceutical Benefits Scheme will pay for. Viv?
Stephen, I just wondered what the experience is in Europe of the treatment approach of boosting macrophage activity through checkpoint inhibitors.
Well, that's a very
Specific question.
very in which disease did you say? Sorry.
DLBCL. Michael can translate the question.
Large
Yeah,
cell.
yeah.
So I think she's referring to the Stanford CD47 SERP1 alpha
Okay.
data in NEGM.
Okay, so I have limited experience in with this PD1, so I will clearly not have any feedback that can provide you. These are too specific. I mean, we didn't have this experience.
It's a very, very new emerging technology that Viv's referring to, which is there was a paper published this year, or just before ASH, I think, late last year, of a way of harnessing the immune system. So the immune system has multiple effector cell types, cells that do the work of the immune system. And we talk a lot about harnessing lymphocytes, but this particular paper was interesting because macrophages, which are the big eaters of the immune system, were switched on by this particular treatment, and it appeared to be well tolerated. Now, the data that was presented in that paper was of very few patients, and we've talked before about New England Journal of Medicine. This is the peak journal in medicine, and if something goes in there, people read it as being sort of gospel. And which it isn't. It's sometimes about who you know, and sometimes it is gospel, but it isn't just gospel because it's there. Yeah. And this was a very early data from a phase 1 clinical trial. Unusual to get it published in that journal. But the reason it was interesting was because the drug appeared to be well tolerated and active in diffuse large B-cell lymphoma, and so a number of companies are now exploring that mechanism of action in phase 1 clinical trials. And there's one in particular that's active in Australia at the moment. And I'm not sure, I can't comment on all the sites, but at least at Austin and Monash.
You raise a very important point, is that there are the publications which show some directions for research, and some of these direction will never be used in clinical practice. There is a lot of hope, and this hope disappeared or was not as expected. And but we are living in a world where you get access to a lot of informations and a lot of hope on the website. But it's very complex to switch from one publication, even if it's in the New England Journal of Medicine, to it have been proven can be used. There is a big bridge to cross.
3 to 5 years minimum.
Yeah, at least, yes, and it has been quite short and very fast. But if we go too fast, we take the risk that the scientific demonstration has not been fully made. And at the end, the government will reimburse and pay the drug, while in fact there is no such a high efficacy. And the talk you were given about CAR T is an example. There are a lot of hope for CAR T cell, but will this hope be true in daily practice? This is still questions pending.
Early days.
I guess this is a quick comment, but basically, cancers or lymphoma has been traditionally being treated by chemotherapy, and the rise of all the immunotherapies have come about, especially in the last couple of years. We've had 13 new immunotherapies, and none of them are chemotherapy, have been approved on the PBS. Where do you see the future of treating lymphomas? Is chemotherapy a thing of the past for a lot of these patients? It's a big question.
I don't know what is your opinion, but we have to keep in mind that chemotherapy has saved millions of lives and is a still very active treatment. We're scared about the word chemotherapy because because it's cancer, right? But it's a word and it works. It's a good treatment. It can save a lot of life. Immunotherapy is active, it works, but some immunotherapy are still a promise and absolutely not a demonstration. So we should not jump from chemotherapy is the past to immunotherapy for everyone frontline. And what is immunotherapy? MabThera or obinutuzumab is an immunotherapy, right? And Imbruvica, we mentioned this drug in mantle cell lymphoma, is a targeted therapy, but it's a chemistry project. So it's a chemotherapy. It's a question of wording.
Antibiotics are chemotherapy, right?
Antibodies are exactly. So what doesn't mean sometimes it's more about advertising. I have a new immunotherapy, and the pharma company like this wording too. But today, chemotherapy remain for a lot of diseases standard of care, and will remain standard of care for a long time. I think it's not in the past. It's not that I'm advocating in favor of chemotherapy, but what we want is efficacy first, right, and not a promise of treatment. Maybe that immunotherapy plus chemotherapy will do better than chemotherapy alone. Maybe a mix of immunotherapy will challenge chemotherapy at relapse. Will it be the case frontline? I don't know. In DLBCL, 60% of the patient will be cured with the chemotherapy. Will immunotherapy do better than that? I mean, no one really knows. So we have to be very careful when we make such promise with immunotherapy, targeted therapy, chemo-3 approach, or well, whatever we call it.
I mean, I agree. And I think that what I like about immunotherapies is that, to me, they might offer a way of getting around this finely slicing of different lymphoma types. The idea that as something that harnesses a mechanism of action that might not be as specific to whether you've got diffuse large B-cell lymphoma subtype 5 or subtype 4 or subtype 3. There are now at least 5 types of diffuse large B-cell lymphoma. If immunotherapy can work across all of those, the way chemotherapy often does for the frontline setting, then that may offer a way to handle patients' disease after it relapses, or it may provide a way of getting around this issue of molecular subtyping. I mean, that's my kind of hypothesis. There's no proof of that. So that's an attraction. A disadvantage of some of these novel therapies compared to chemotherapy is that for some of them, tablets, intravenous injections, need to be continued, as far as we know. And one, I think, really important philosophy that we need to remind ourselves of is that, in my view, good quality of life is being cancer-free and being off treatment, if you can. And there's a lot of treatments coming out that require continuous tablet taking or continuous therapy, and that might be a step up on what we've had before, but where I'd like to see us go is to be able to stop those tablets. And there are some, I mean, at ASH last year in CLL, there's some really great data where finally people were standing up and saying the point of this trial was to stop treatment. Thank you. You know, that's exactly what we need to see. And so I think that's what I think the future is.
They definitely don't come without their own side effects as well, of course. Any other questions?
I can talk a little bit about CAR T.
CAR T, yep.
Yeah. So I thought I would just give a brief update on the current status of CAR T in Australia, given all the recent press. As you may know, the regulatory authorities in Australia approved Kymriah, which is the brand name for a cellular product also called Tisagen-Lec-Lucel, which is manufactured by Novartis. And in November last year, the federal government basically took that first step to acknowledge that this product was deliverable, that it clearly had evidence of activity, that it did what it said it was doing. And so they basically said, yes, this drug can be marketed in Australia. That happened in November. And then shortly after the federal election was called, the health minister announced approval of funding for this cellular product in acute lymphoblastic leukemia in people up to the age of 26. so up to the age of 25 and no older, and acute lymphoblastic B-cell leukemia. So this is similar to lymphoma in the way it presents itself to the immune system. Now, CAR T cells are a platform, and so when someone says CAR T cells, it doesn't. It's a bit like the word chemotherapy. It could mean many different things. It doesn't refer to a specific target or a specific disease indication, or necessarily a specific product. And so one CAR T cell product may not be the same as another CAR T cell product. And so this approval was not for all CAR Ts or for all cancers, but the specific use of a specific CAR T in a specific indication. To explain what CAR Ts are, many of you will have seen this information before, but just to sort of revise, essentially what CAR Ts are is a designer immune system. So we take a branch of the immune system, these effector cells, in this case T lymphocytes, and we train them to target the lymphoma and give them back to a patient as an infusion, which is given as a one-off dose. When the T cells see the lymphoma, they expand and stay alive in the body and continue to actively kill the lymphoma. So it's a one-off treatment that is live. It's a live therapy. So the way it actually works as a mechanical thing, this is a Novartis slide, is that we collect the lymphocytes using a procedure that's a bit like a stem cell collection. It's called leukapheresis, and it involves sitting in our apheresis department, having a line taking blood out and a line returning blood, and we spin the blood in a centrifuge and take out just the lymphocyte layer, and we put those into a bag, and in this particular case we cryopreserve it in liquid nitrogen and then send that container to a manufacturing facility in the United States. There in the United States, they use a virus to make the cells develop a new protein on their surface, and I'll show you what that protein looks like. But basically what the protein is it's a chimera. It's why it's called chimeric, and the idea is that it harnesses the power of kind of vaccine-type responses, so the power of antibodies, and combines it with the power of cells to kill into a single thing. So you've got this kind of mutant cell which is designed to specifically target the lymphoma. And CAR Ts can target their target irrespectively of what else is going on in the immune system, we think, and don't require all of the signals that are usually required for T cells to attack and kill their targets. So they're quite a clever treatment, very targeted. They're trained up to target a specific thing. So in this case, it's CD19. Just like CD20 is on the surface of lots of B-cell lymphomas, CD19 is on the surface of lots of B-cell lymphomas. And so this theoretically might be applicable across a range of B-cell lymphomas that bear CD19, but the clinical trials have, again, just been in some of the more common diseases or some of the more kind of intractable areas of need, such as relapsed pediatric leukemia. Now, so once the cells are made into this product, we give just a little bit of low-dose chemotherapy and then give the cells back as a single infusion. Here at Peter Mac, we've set up a CAR T service to deliver this treatment, and that's largely because we're running clinical trials, but we are now delivering the treatment for patients with acute lymphoblastic leukemia in particular, pediatric patients according to that government approval. And here we have a referral process. A referral comes in, reviewed by a senior doctor with a specialist interest in the disease in question. Patients are then enrolled into a CAR T program. We organise the apheresis collection, and then give the infusion. Now, for the particular one, Kymriah, we generally manage patients as an outpatient and then bring them in if they've got any kind of toxicity, which occurs more often than not, for sure. And we ask patients, and this is a question for regional rollout, we ask patients to stay within half an hour, ideally, of the hospital so that we can manage toxicities as they arise. Now, Kymriah was also assessed in diffuse large B-cell lymphoma. So I won't go through the data in much detail, but just to say a clinical trial has been done in diffuse large B-cell lymphoma, and the company that makes Kymriah has submitted to government for approval for funding in diffuse large B-cell lymphoma, and the Medical Services Advisory Committee has scheduled to review that in the coming months. There's a second company which has made a similar submission using a different CAR T, but also targeting CD19, that's also being assessed by the federal government. This particular trial was run globally, and Australia was involved in the trial. The single most common side effect of CAR Ts, they're not free of side effects. They can be pretty rough on a patient, or they can be well tolerated. And we're not very good at telling which patient will have a rough time and which patient will have will find it relatively straightforward. But this syndrome called cytokine release syndrome is the biggest key side effect. And basically what it is the CAR Ts see their target, they expand, and when they expand, they release hormones. So from a patient experience point of view, it's a bit like having an infection. And so fever is the single most common side effect of CAR T reinfusions, but that can then go on to become a bit like a serious infection or serious sepsis in the way a patient's body reacts as the CAR Ts release hormones into the body. And so a fever can then become a fever with low blood pressure or a fever with low oxygen levels, which requires intensive care support. And we have antidotes to this that we use to target this, but it all needs to be done in an environment where people are used to looking after this syndrome. Because some things that we might use to treat infection or we might use to treat low blood pressure, we don't want to give to patients who've just had CAR Ts. So understanding that distinction is one of the things that's important. And just as we talked about before, we probably need centres that are giving enough of this kind of treatment to be doing it. It's not something we're going to be seeing given out in a distant regional hospital routinely, not at least in the short term. The different CAR T agents do differ, even if they target CD19, they do differ in terms of the rates of toxicities, in terms of what's been published, and so the specific product that's being given probably does matter. At the moment, there's just the Novartis product available outside of clinical trials. But it's clear that this toxicity of cytokine release syndrome does seem to vary between the different routinely available products in the US and Europe. The other key toxicity is confusion, which can lead to severe confusion requiring an anaesthetic for a patient to recover from. So you develop confusion, we may require some sedation to manage a patient who's very seriously confused and need to get through that period and recover. So most patients recover very, very well from that. But again, a patient needs to be physically fit enough to be able to tolerate these insults. So I don't think CAR Ts are going to be treatments that we easily give to very frail people or people with other serious medical conditions, because what we wouldn't want to do is give a treatment like this and then for the side effects to shorten someone's life. And so, I mean, we face this kind of a dilemma with every treatment we give. CAR Ts look to be active, but may not be simple to deliver. Broadly speaking, I've mentioned that there are two companies bringing CAR T into routine care in Australia. In addition to acute leukemia, the only lymphoma indication under current consideration is patients with diffuse large B-cell lymphoma who have relapsed after 2 prior therapies or autologous stem cell transplantation. We don't know whether the funders in government are going to put on additional restrictions in terms of CAR T, so we just have to wait and see. About 40% of patients, so a significant minority, have a long-term good outcome from CAR T therapies. So a lot of people think of CAR Ts as being the silver bullet or this magic treatment because it's high-tech and because it's expensive and because it's custom-made. But in fact, it isn't. It's a good treatment. It's better than the other treatments we have, I suspect, for patients who've had several relapses of their diffuse large B-cell lymphoma, but it's certainly not guaranteed to work. And again, the toxicities probably do vary by the type of treatment being given. Now, there are a number of active clinical trials in Australia. There's been some press recently about Westmead Hospital, and they're developing up a CAR T that also targets CD19, but it has a different manufacturing process. And that's looking at particular lymphoma subtypes again, and it's just getting going in a phase 1 trial. In follicular lymphoma, there's a study for patients who've had several relapses of their disease that's running at the hospitals that I've listed here, open here at Peter Mac, but also in Brisbane and in Sydney. One of the critical questions that we need to know about is CAR T better than autologous stem cell transplantation for patients who've relapsed and have a particularly high risk of not doing well after an autologous stem cell transplantation? And so there's a randomised trial of CAR Ts versus autologous stem cell transplantation in patients who've had their first relapse of their diffuse large B-cell lymphoma within 12 months of original diagnosis, which is currently active here and which is likely to be active at other sites listed in the coming months. We are exploring CAR T for mantle cell lymphoma, chronic lymphocytic leukemia, and we have several phase 1 trials looking at essentially focusing on diffuse large B-cell lymphoma in the short term. So for other lymphoma subtypes, such as marginal zone lymphoma, we don't yet have CAR T. And to my knowledge, there's no CAR T program on the horizon for Hodgkin lymphoma in Australia at the moment. An area of real challenge is whether we can find targets for these CAR T platform beyond lymphomas. We have active clinical trials in solid tumours, but there isn't yet a big hit in that space. There's several hundred CAR Ts being tested in early phase clinical trials around the world. Probably the next disease where CAR Ts will come into routine use is myeloma. I suspect, that's my guess. But at the moment, the only 2 approvals for CAR Ts globally is this paediatric ALL population and patients who've had several relapses of their diffuse large B-cell lymphoma. And hopefully Australia will come on board in terms of being able to provide it, but it's an extraordinarily expensive treatment. It's custom made, it's $375,000 just for the product, not for the care and the associated hospitalizations. So it's a real challenge for our insurance system to bear that cost. At the moment, the Australian government is supporting individual patients to go overseas to receive this treatment while it's in negotiation with industry in Australia. So there's something called the Medical Treatment Overseas Program, which is open for treatments that are approved overseas but not available in Australia. And so several patients with large cell lymphoma have gone to access CAR Ts that way in the last couple of months. The other thing I should mention here is that Peter Mac is currently, this is in the public domain, is in negotiations with Novartis to become a manufacturing site for CAR Ts, which will hopefully shorten the time to access for these treatments. And that will only, of course, happen if there's a way for patients to fund them. So it's a lot of big challenges for the companies trying to bring these treatments and the doctors trying to bring these treatments and the regulators. Greg Hunt announced that Peter Mac would become a national centre of excellence for CAR Ts and announced a major grant for Peter Mac in recent months, and we're going to use that to try to expand our clinical trials in CAR Ts and make CAR Ts more available to patients. We won't be using it to treat patients just for routine things, but we'll run carefully controlled small clinical trials to try to improve which patients can receive CAR Ts and perhaps improve the way CAR Ts work. So that's our program at the moment. I should just mention that we don't know that CAR Ts are the be-all and end-all. There's a whole bunch of other new therapies floating around at the moment. In Australia, there's a clinical trial of these CD20, CD3 bispecifics. There's a trial here of a particular one, and it's also open at Prince of Wales, and that's a Roche bispecific. And then there's another trial of a very similar agent at multiple hospitals around Australia, and that agent is also made by Roche, Roche Genentech. And basically what this does is it tries to mimic what CAR Ts do, that is harnessing lymphocytes to kill lymphoma, by this time forming a physical bond, taking Gazyva and basically binding it to T cells. So you've got a double-headed antibody that forces T cells to attack the lymphoma. And so this particular mechanism of action is currently in clinical trials, and it may all turn out to be, as with all clinical trials, we have to be very reserved about talking about active clinical trials. But just to say that these ways of harnessing the immune system are under active exploration. Cells may be part of it, but they may not be the whole story. Right. Coming
Thank
up
you,
to
Michael.
20 to 12.
We're doing very well. Excellent. So if patients have any questions about how to be referred for or more information about the CAR T, how do they go about that?
There's an email address, car-t.inquiry@petermac.org. So on our website, if you didn't hear that, it's there's a link on our website, so you can just email inquiries. Our approach has been that just like any other referral, we like your haematologist to be involved. We don't generally give out individual patient advice. We'll give kind of key frameworks about, yes, this might suit your particular disease, but we'll always ask that a referring haematologist is involved.
Yeah. Excellent. Is there any questions? Oh, yes. I'll start over here.
The issue of cost has come up quite a fair bit today. I understand that the Westmed Institute, they're working on the CAR T as well. The cost is significantly lower, and I think it's about 50,000 versus the 500,000. Is that the same? I know you've mentioned there could be differences. Is it different? Like, is there different CAR T
It's
therapies?
a different construct targeting the same target. So its philosophy in terms of its mechanism of action is exactly the same. But these CAR Ts differ in how they're manufactured. The receptor on the surface of them varies significantly, and we know for sure that makes a difference in toxicity. There are some CAR Ts where there's been a lot of toxicity and it's been unacceptable and they've not been able to move forward. So the Westmead one's at its very earliest phase of development. It's certainly a good idea. We collaborate with the Westmead. We'll be running their trials here too. But the key issue is the trials need to be done. And as Stephen said, trials are expensive. They have a massive overlay. And a lot of that overlay in terms of regulatory requirements then feeds into what the final price will be. And so there's a difference between cost and price. And the price, I think for any of these things just remains to be seen. I think it's nice to hope that a home brew will be cheaper to patients and to the government, but the government's not going to soften its regulatory requirements about paying for anything. Until the hurdle has been leapt by a particular treatment. It has to be shown to be safe and effective. And whether it's $50,000 or $500,000, you want to know that you're getting what you paid for. And that bit about knowing that the product that you're buying is the product that you're buying, that when you buy an aspirin, it's an aspirin and not a Panadol, and that it isn't, diluted by other things, and the company's, keeping its quality assurance, that is a big part of the cost of CAR Ts. So I saw the 60 Minutes story too, and we, as I said, we work very closely with that team and like what they're doing. But I think it's too soon to say that there will be that treatment will come to market, and B that the costs will be something that is routinely reimbursed by government. So I think we have to play every strategy and hope that with time the price of everything comes down.
Michael, I've got lost in the fact that we give a modified T cell for a B cell lymphoma. There's something in the logic there that I'm not seeing.
Yeah. So the answer to that is the T cells are the killer cells in our immune system. a bit like the question around macrophages, which are another type of cell that we're not yet very good at harnessing. T cells we kind of understand. And the philosophy, what we learned is that if you take someone's entire immune system and put it into another patient, allogeneic stem cell transplantation, that the T cells in the allograft probably have to do with the graft versus disease effect. And so this is just trying to harness that effect, increase the specificity for what's hit by forcing the cells to target something, and that increases the sort of benefit to toxicity ratio. You don't get graft versus host disease because you're using the patient's own cells. In this particular example, there are other examples where we don't. So so that's the idea. Now the fact that we're targeting B lymphocytes just happens to be the thing that's first off the rank. But we've got a CAR T cell program here that targets a protein that's in other cancers. It just so happens that this particular CD19 just seemed to be the first proof of principle, and B lymphocytes are like any other tissue philosophically in that they can be targeted. The question is, why this was the first off, I don't know. Probably liquid tumours are easier to target than solid tumours. But it could easily have just been a myeloma target or a lung cancer target or something else that got there first. It just so happens this is the first one.
Okay.
Do you have CAR Ts in France?
Yeah, we do. It's approved. We have 2 drugs.
Yeah.
So it has been approved last summer
Yeah.
for a prescription. But it takes time for the centers to get approval by the company itself.
Yeah.
It's not only you get rights to prescribe, the company give you the authorizations to use the drug, and they have the internal process, which is different from Novartis to Gilead. So it's also a lot of time. So we get access. We have 10 centers that are open for CAR T, and we started the program last summer in Nantes.
What's the population in France?
You mean the population of the patients, or
Or the whole country? France.
Oh, it's 64 million.
So 64 million,
Yeah.
10 centres.
Yeah.
How many
So
centres
we
does
expect to
Australia
treat
need?
400. That
Yeah.
was for the LBCL.
Yeah, 400.
400.
Okay.
But I'm sure we're going
Similar
do
to the
more than
Australian
that.
estimates. Yeah.
We treat one patient every 2 weeks in Nantes, so I guess there will be more than 400 French
Yeah,
people
400 sounds
start
like a
to get
major
it.
underestimate. Yeah.
But I agree with you, what you said about the cost and the price. It doesn't cost that much to build CAR T cells.
No.
It's not right to say that. It's a price, but it has nothing to do with the real costs to manufacture these things. You could do it really for low price in the lab. CAR T are known for 30 years now, and it works in clinical trial because of the 4-1BB translation. But it's a major problem. I think this cost, this price has to be discussed before to move on.
And it's just going to be a question again around, I think sometimes having a lost leader, we can sit back and not take on these therapies and wait for the price to come down but then our patients won't get the treatment. So to some extent, the countries that are taking on this treatment are agreeing that for a while it will be a lost leader, with the expectation that market forces and competitors like the Westmead product and those sorts of things will just force the price down, just like any other drug. Now that's not always worked. It doesn't always work in the American environment, but it probably will work globally, we hope. Yeah.
It becomes more common. All right, I think with time, we better finish things off. Look, I'd like to, on behalf of everyone, just like to really thank Professor Gouill for coming to Melbourne and speaking to us all, and of course Michael Dickinson, as always, for helping support today's event. And thank you all for coming today. It's I know getting here on a Monday morning through Melbourne traffic is always a bit of fun, so I really appreciate your support. And if there's any further questions, you can always give me an email on the inquiries line or call the nurse support line, and I can find those answers for you as well. So I'll join me to thank our guest speakers. Thank you.